into the webinar series that we have been conducting since 5th of June. And the webinar series we named as first outcome-based biotechnological research and education, a roadmap. So this is the 5th webinar under this series we are conducting. So far, the first one delivered by Dr. Alok Das from IITR Kanpur, then Dr. Bibha Ahuja, Chief General Manager, BCIL, and third one was delivered by Dr. TJV Higgins from CSIR Australia, and then fourth one was from Manoj Prasad, Dr. Manoj Prasad from NIPGR New Delhi. So this is the 5th one, which will be delivered by Dr. Adam Price, our collaborator in RISE research. He collaborating with us for quite long years now, maybe for the last 15 years or so. And the other is from University of Aberdeen. He's a very senior professor there now, and he is a world-known rice geneticist. So I would like to, he has been collaborating with our RISE group led by Dr. MKMT in our center. That's why I would like to request Dr. Modi to make a brief introduction of Dr. Adam Price. Thank you very much. So please, Dr. Modi.
Thank you, Vidut. It's my pleasure to introduce Professor Adam Price of School of Biological Sciences in University of Aberdeen, United Kingdom. As you told, he is a renowned geneticist and a very keen photographer. Not many of you might be knowing this trait about him. He is a very keen photographer. And he is, I think, the only person that grows rice in that area. Because Aberdeen, you cannot grow rice in the outside. He grows rice there in the greenhouse so that he can work on rice. He loves rice so much. His current research interests are Qtl and association mapping in rice, or drought tolerance, biotic factors like blast, striga, etc. Also about nitrogen use efficiency. And he has developed a association panel called the BAP population, and he will be talking about his work on this BAP population. BAP means Bengal, Assam, Ost panel, and he will be talking about that. So I welcome Dr. Adam Price to this webinar series and request him to give his talk. Thank you. Adam, please.
Thank you, Professor Modi and Professor Bijat, for inviting me to speak to you today. It is a strange circumstances. As has been introduced, I have been collaborating with Assam Agricultural University for many years. I think 5 or 6 times I have visited, and I've done probably 3 talks already on related subjects. But what I want to do today is give you an update of where we're going with our new association panel. By way of introducing it, I'll give some background on some basics about genetic mapping in rice. Yeah, so what I'm going to do is I will share my screen now and take it from there. So hopefully you are now seeing my slides. Can someone confirm that's the case?
Yes, the slides are visible now. Yeah.
Okay.
Yeah, it's visible now.
Okay, so this is what I want to talk to you about, the Bengal and Assam House Panel for Genetic Diversity and GWA Mapping Studies in Rice, which is more than anything a piece of work that has been conducted by myself and my colleague Gareth Norton at the University of Aberdeen with a large number of collaborators, and including people from Assam Agricultural University. So now I don't know how to change slide. That is not working. That is a shame.
You should be able to change the slide normally, just like.
Normally you change.
Just
Yeah.
go to the slide.
That is not changing slides, so hold on. I've got two screens
Yes.
open. I guess this is, did that change slide? No. Okay, I'm gonna stop sharing and I'm gonna share on a different screen.
Now it should work. Adam, Rupjuti, can you tell how to change the slide?
Sir is using two screens, I guess.
Oh, don't use two screen. In your own screen, you just share the slides. Yeah.
No, it's changing. Stop sharing. This might take a second.
Yeah, here itself you share the slides. From your side, if you go to slides there, share screen. You share your screen. Yes, it's coming now.
Yeah, but now
Yes.
my arrows don't work. Why on earth is that? What can you see now? The most northerly rice field.
Yes, sir. We can see it now.
Slides. That is odd.
Yeah.
I'm going to change the way I'm doing it. I can't do it on projection. I would just stop the projection a second. I will just show you the screen in a different way. Okay, so I'm going to do it this way.
You can put in
What are
the
you seeing
slides.
now?
Sir, just put it in the full screen mode now, sir. Then
Full screen slideshow mode.
yes, sir.
Yeah, it's okay. It's okay, Karan. It's okay. Adam, it's okay. It's coming now. Go ahead.
So you see the most northerly rice field in the world. The world's most northerly rice field.
Yep.
Yeah, sorry about that. Okay, so yeah, as Mahendra said in his introduction, we work on rice in a very northerly part of the world. But we can do that because we can grow rice in a greenhouse and we heat it to 25 degrees. But I'd like to convince you that by collaborating with other people, we can do work that is highly relevant to rice breeding. So this is the format of a talk I want to give. Ultimately, I want to talk about the Bengal and Assam House panel. But I first of all need to introduce why we felt there was a need for a new association mapping population. And then I will talk about the development of this new panel, some results, some sort of novel aspects to the way that we analyze the data by aligning it to a different reference. Say something a little bit about what else we're doing in addition to the main experiments that were used to fund this project development, and some concluding remarks about the achievements we have made and some of the challenges we yet face. So first of all, why do we need a new association panel? Actually, there are a lot of them around now. So it's becoming a very, very popular method of research. But I started my work, my life as a rice geneticist on a conventional biparental cross population, which was a cross between the rice variety called Bala and one called Azucena. And that was 25 years ago, more, that we developed this population. And we've used that to look at a whole load of traits. I'm not going to go into any of these in any detail at all. I just want to highlight one thing, is that one of the traits that we were interested in, even from relatively early days, certainly for the last nearly 20 years, is grain arsenic, because the accumulation of arsenic into the grain of rice is a problem in those parts of the world where there's a lot of arsenic in the groundwater or in the soil, and that happens to be especially a problem in Bangladesh and West Bengal. So from that population, the conventional cross, we have more than 30 papers describing quantitative trait loci, QTLs. So that is really the main subject of what I do my research on over the last virtually 30 years, Qtl mapping in rice. And when we do that, as a biologist, I'm always interested in what is the gene underneath the Qtl. You can research Qtls for a number of different reasons, and I guess the best justification is that they can be useful in breeding. So for example, in Jharkhand, there's a rice variety been bred with deeper roots through Qtl work we've done on the Bala Azasuna mapping population, just using marker-assisted selection. But as a biologist, you're interested not just in the application of your Qtls, but what are the genes under it. And this highlights why GWAS is particularly interesting, because the next slide tries to give you the background about accuracy of Qtl mapping. 2006, I wrote this article, which is really just summarizing the fact that Qtls, this is before association mapping was ever possible in plants, using biparental crosses, they are reasonably accurate. But this slide, which comes from this figure, which comes from that paper, tries to illustrate the point of accuracy with Qtl mapping, conventional Qtl mapping. So what this is looking at is where we take traits of plant height in the Bala Azasuna mapping population under drought conditions, control conditions, average of all conditions, low nitrogen, low light. And we map them, on, in this case, on the bottom half of rice chromosome 1. And this is the test statistic, the LOD score. The higher this is, the higher is the probability there's a Qtl there. And you can see for each of these traits related to plant height, you get a Qtl around 170 centimorgans in our population. And because we have access, even in those days, we had access to the genome, we can relate the genetic position there to physical position. And we know that the gene responsible for this Qtl is called SD1, semi-dwarfing gene 1. So we can actually see how accurate our Qtl mapping is. And it's not very accurate in actual fact, because within a space, let's say from here to here, you may be looking at 400 or 500 genes. So if you didn't know which gene it was, you'd be hard put to discover it just by mapping alone. You'd have to have some extra information, or you have to go through a very long-winded fine mapping approach. And that desire to get closer to the underlying gene for a Qtl led our group to focus a lot on association mapping. And the first piece of association mapping we ever did was a collaboration with Cornell University, Susan McCutcheon Cornell University, who was developing, who led a project, a collaboration developing this thing called the Rice Diversity Panel, which collected rice cultivars from around the world, 82 different countries, 371 different rice cultivars, and at the original stage they had 44,000 markers. Instead of the 164 markers we had in our mapping population, they've got 44,000. So association mapping is a technique that is possible because of the relative ease and low cost of using SNP markers to characterize material. And as those markers developed, association mapping developed alongside it. So 44,000 markers. We grew those in Aberdeen, we grew them in Bangladesh, and we grew them in China. And I'm going to show you some quick results about that. But that population, the first paper on genome-wide association mapping of that paper actually used some of our data. So we were authors. And I say something about that panel, that population, as an introduction to some basic genetics of rice. So the map at the top there shows you where they collected their cultivars from. This was a collaboration between Cornell University and the International Rice Research Institute. They collected their rices from all over the world, but they used the markers to classify the cultivars on different subgroups within rice. So there are recognizable different subgroups. One is called indica in red, one is called aus in yellow, one is called temperate japonica, and one is called tropical japonica. And you can see from the map where these are coming from. And actually, particularly interesting to this audience, and also highly relevant to this talk, is the fact that sitting right on top of Bangladesh and overlapping with Assam are a whole load of cultivars which are yellow from the Aus subpopulation. This is the results of one of the traits that we helped them measure. We measured flowering time in Aberdeen and measured flowering time in Faridpur. And this is the way that these QTL results are presented for those who are not familiar with association mapping. So here we have the 12 chromosomes of rice along this axis. And on this axis, we have a test statistic. And each dot is a different marker. So there should be 44,000 dots on here. And the higher the dot, the higher is the statistical significance of the association between a marker and the trait. So for example, up here, you have loads of SNPs, loads of markers with relatively high values. That means that there's a high significance to the association of these SNPs with flowering time. And that's interesting in a biological sense, because actually just on top of those SNPs is the gene called HT1, which in a historical sense is interesting because it was the first ever cloned QTL in rice, known to affect flowering time. But this highlights, this slide actually highlights some really important features of genetic mapping. First of all, you can see that in contrast to the genetic mapping I showed you before for plant height in the Bala Azucena population, these peaks are relatively narrow, which means the number of genes underneath the peak are relatively small. Another thing it shows you is that if you do the same, measure the same trait in one place and you do it in another, you'll get a different result. So for example, on chromosome 12 is a really strong QTL for flowering time measured in Faridpur in Bangladesh, but it's absolutely not apparent in Aberdeen or in Arkansas. This is a feature that people working on QTL mapping know very well, QTL bioenvironment interaction. But you can see it very clearly here. We worked on that population largely because we thought it would be a tool to allow us to get QTLs for grain arsenic. So we measured grain arsenic in that population when we grew it in China and in Bangladesh. And we collaborated also with a group in the USA who grew it in Arkansas and in Texas. But we when we did that, we could easily identify a major problem trying to use this panel, this population, to look at variation in grain arsenic. And that is, when you look at the amount of arsenic in the grain of rice, for example, in Arkansas, you can see that it is strongly affected by flowering time. So here, the plants that flower late have higher arsenic than the ones that flower early or in the middle. I haven't actually said why we're interested in arsenic, have I? So I would better mention that. Arsenic is a class 1 carcinogen, and rice just happens to be very good at taking it up, both into the shoot and to the grain, probably because rice is also adapted to take up large amounts of silicon, and arsenic in the arsenite form is an analog of silicon and is transported through silicon transporters. So rice just happens to be good at taking up arsenic, including into the grain. So what this slide shows you is you get an interaction between flowering time and grain arsenic. And unfortunately, it's not consistent. So this is Arkansas in 2007. This is Texas in 2009. Again, a strong association, but a completely different pattern. And what that meant to us was that this panel is possibly not brilliant at measuring or discovering QTLs for grain arsenic. So flowering time massively affects grain arsenic concentration. I could go into exactly why we think that is, but I'll take questions on that if you're interested. Okay, so we did use this population, and we did publish a paper on grain arsenic mapping in this population. And this is the slide which summarizes that. It's a fairly horrible slide. I've shown this in many talks, and I always apologize for the amount of information that is presented on this slide, and to myself really for the fact that it doesn't show anything that is in any way exciting. But I need to explain what this slide is for a moment. I might do that by popping back to this. You see these slides, I told you, the way that these plots, we're called Manhattan plots, the way they tend to be, the way they tend to occur is that most of the SNPs have very low p-values, and they cluster down here. So in the slide I'm going to show you and talk to you about in a minute, we've cut it off here at 4, and all the ones below 4 you can't see, just because they make the slide more messy. So if I show you this slide now, you notice that the scale starts at 4. So all of the SNPs that are not very significant are not visible. Only the ones that are over 4 are visible. And what you see here is QTL, or SNPs that are associated with grain arsenic, color-coded. So they're color-coded by the site. So you probably can't see the writing, but say, for example, red are Arkansas in 2006. So these red ones, Arkansas 2006. And black are Arkansas in 2007. So the different colors represent different sites, and there are 5 different sites. And the other thing that you see on this slide is that you have triangles and circles and crosses. I mean, that is because when we use this population, we do multiple GWAS analysis. The crosses are what we call combined analysis, which takes all of the data of all of the cultivars for which we have data and do the analysis on that. But for example, the circles are just the ouses, the squares are just the indicas, the triangles are just the tropical japonicas. So it's very complicated, the analysis we're doing. And there's a very simple conclusion from all of this, and that is that there's almost nowhere on this mapping exercise where you see QTLs for grain arsenic which occur in the same place from different experiments, or even from different subgroups of the population. And the different subgroups of the population we think is very important, because there are some serious complications about doing GWAS when you have strong population structure like you do in this population. So we decided that we needed a better population for doing this analysis. So that was the justification for writing a grant proposal, which got funded in 2011. So it's 9 years ago we started this project. And the project had a very long title called A Genetic Dissection of Traits Required for Sustainable Water Use in Rice Using Genome-Wide Association Studies. So it's a horribly long title, funded by the BBSRC. And it was a collaboration with several institutions. And I will mention those in a second. The objectives of the project were the main justification was that alternate wetting and drying is a promising water-saving technique being promoted in Bangladesh, but we don't know if cultivars and genes are optimized for it. We don't know how it improves yield. So if you go around Bangladesh and talk about alternate wetting and drying, everyone says it improves yield. We don't know why. We don't know if it's sustainable. And we really wanted to use this project to get a handle on the genetics of grain arsenic and possibly grain cadmium as well. I'm not going to mention cadmium. So the partners in this project it was led by Aberdeen. So I was the leader, but we had several people in Aberdeen. So Andy Mahag is now in Belfast, and David Salt is now in Nottingham, and Oliver is now in Dusseldorf. So there's only Gareth and I left from the original starters of the project here. But we had a collaboration with Lancaster on physiology. Erie helped us substantially with, especially with the making the population, selecting the cultivars. And most of the main experiments were in Bangladesh Agricultural University. But we had a collaboration also with Calcutta and with Assam Agricultural University. And you see Ram and Sama there. And I need to maybe just say a little bit about what alternate wetting and drying is. It's a technique, as I say, being heavily promoted in Bangladesh. Here's a leaflet made by the International Rice Research Institute in Bangla. The idea of AWD, as we call it, is it saves water. The farmer takes a little tube like this, which has got holes in it, places it into the field, so they can look down the tube, and then they can allow the water level in the soil to drop until they can no longer see water in the tube, and then they flood it again. And by doing that, they save water, but the soil goes through cycles of being aerobic and then anaerobic, aerobic and anaerobic, rather than being anaerobic all the time, which is conventional flooded rice. So this project had a lot of expected outputs, and I'm not going to talk about many of them. I'm just going to highlight two of them today. That is, one of the objectives was to make a collection of genetically characterized ouse cultivars for Genome-Wide Association Studies in Bangladesh and Northeast India. And the other was identify candidate genes for adaptation to alternate wetting and drying, and for the uptake and transport of macro and micro elements. And all I'm going to talk to you today about is arsenic. So why ouse? Because this is a sort of phylogenetic tree, a neighbor joining tree of the genetic diversity in rice. And rice falls into two subspecies, that is indica up here and japonica down here. And within japonica, there are two recognizable subgroups, temperate and tropical japonica. With indica, there are two recognizable subgroups. One is called indica and one is called ouse. They originate from Northeast India. So we were led to believe when we wrote this proposal. Many, possibly even say most, of the current donors of abiotic stress resistance are ouse in origin. So FR13 is the donor of submergence tolerance. Dular is a drought resistant variety used in breeding. Black Gora, we use this quite heavily because it's our deep rooted Czech. There's a variety called N22, a lot of you will be familiar with because it's used as the heat tolerant Czech variety. So a lot of these ouses show strong tolerance to abiotic stress resistance. Quite recently, the group in Cornell did this really nice study of genetic diversity of Rhiza rufipogon, the wild progenitor of rice. It's a beautiful paper, actually. And what they conclude is W4 is a recognized subpopulation of wild rice that comes from the east coast of northern India, Bangladesh, down into Myanmar. And you can't see this here on this slide, but this is that group in orange, and in yellow is ouse cultivars. So this is the wild relatives of rice that are strongly related to ouse. So this is where ouse comes from. And they say in their paper, with its unique geographic, genetic, and ecological history, the cultivated ouse subpopulation and its wild relatives, W4, this group here, represent an underappreciated genetic resource. So it's widely recognized that ulcers are particularly interesting in rice genetics. So we started off this project by using a small number of markers and characterizing about 500 different accessions that we suspected were AUS. So this is a principal components analysis of those 500 cultivars which we suspected were AUS. This is principal component 1 versus principal component 2, and what that principal component does is it pulls out indicas and japonicas from AUS. So some of the cultivars we suspected would be AUS turned out to be indica, some of them turned out to be japonicas, but a lot of them turned out to be AUS. And within that, principal component 3 pulls out 2 different subgroups of AUS. Before we wrote this paper, there was only 1 recognized subgroup of AUS. So we said that there was probably 2. And that paper describes that in quite nice detail. There's some quite nice stories about that differentiation of the 2 AUS types. So that led us to produce, from the 500 that are in this study, we selected 300 to make our panel. And there are important features of this. Of these 300, mostly AUS landraces, and mostly they flowered at the same time. That was assessed in the International Rice Research Institute in the Philippines. We had data on flowering time, so we could make them, try to select them that had similar flowering times, at least in the Philippines. In addition to the AUS cultivars, we had 30 or so non-AUS Czech genotypes. So those were first grown in IRI, and then the DNA was sent to us. We had them sequenced in the Genome Analysis Center. I've forgotten its name now. It's changed its name. Earlham Institute, I believe it's now called. So we got them sequenced to about 4x, 5x coverage, all of those cultivars. 4.5 was the average, 300 accessions sequenced. And we aligned them to Nippon-Bari reference to give us 2 million markers, 2 million SNP markers. They were grown in Bangladesh Agricultural University under either continuous flooding or alternate wetting and drying in 4 replicates over 2 years. And a team of people was needed to get those out into the experiment. And the leaves and the seeds were sent back to Aberdeen, and we used ICP-MS, this machine here, to analyze elements. All of the elements in yellow we have data for. I'm only going to talk about 1. If I had hours, I could talk to you about many of them, because we got some excellent results, as well as the arsenic results. We got some excellent results for cadmium and for iron, and we haven't really spent enough time looking at the rest. And for sodium, actually. So to get that data, we had some technicians doing masses and masses of grinding and digestion. We had bioinformaticians, we had statistical support for spatial analysis, and we had experts in GWAS analysis. So it's a really big team that is responsible for producing the results that we did get. And I'm now going to show you some of the results. So the first paper on this mapping population, I won't go into much detail on it, but I'll just show you 2 pieces of information that we published in the first paper. First of all, the flowering time when we grew this in Bangladesh, 83 to 94% of the AUS cultivars flowered within a 10-day window. So we were very happy with that. We managed to do something we think is really important for any trait related to grain traits that might be influenced by flowering time. They all flower, most of them flower within a window. And we recognize, using those 2 million markers, that there are actually 5 subgroups. We think there are 5 subgroups within rice, within AUS. Interesting, some of them are specific to Bangladesh and some are specific to India. I forget which ones. Some of them are found in both India and Bangladesh. So this is where we got to with arsenic. So this was published only 6 months ago. This is the Manhattan plots for grain arsenic. As I said, we had 2 years, year 1, these two, and year 2, these two. We have alternate wetting and drying and continuous flooding. So we have 4 different grain traits, and we have these results, which to the untrained eye look complicated. Actually, indeed, they are complicated. But what I'd like to do is to take you through just 2 of these peaks. So, for example, we can detect 6 QTLs which are detected in multiple years and multiple treatments. So these are 6. And I'm just going to take you through 2 of them. I'll start with this one. So this is the top of chromosome 3. We appear to get a peak in 3 out of the 4 years. Doesn't seem to be a peak here in CF year 2, continuously flooding in year 2. But in the other 3, we do get a peak. And in the paper, we produce this figure. So I'm going to take you through this quite slowly. So first of all, this one. This figure shows you all of the SNPs which are significant in a very small area around this QTL. So we're looking at a really small region, 270 kilobase region, really quite small. And then all of the SNPs that are significant, color-coded by which trait. So in blue, you've got continuously flooded grain year 1. And in green, alternate wetting and drying year 1. So you can see there's a black one there, so that's continuously flooded year 2. And there's some red ones here. So we see that's a sort of a zoom in on that locus. And then we have this plot of local linkage disequilibrium decay. If I had longer, I would take you through this more slowly. But critical in the assessment of where a gene is underneath a QTL is an understanding of the linkage disequilibrium decay. So this is a measure of how far away from the gene you would have to, from the markers that are significant, you would have to go to be reasonably confident that your gene is in that region. So this is a plot of local linkage disequilibrium decay. We tend to use this as a cutoff. We go, this is a R-squared value, we go down here, and we say local linkage disequilibrium decay is 364 kilobases. What that's saying is the gene, almost certainly, that is responsible for this QTL is within 364 kilobases of the peak SNP. Most likely quite a lot closer, but not further. We take all the SNPs that are in that region, and we do a cluster analysis. And when we do that, so this is a cluster analysis based only on the SNPs within that region, and we identify 3 clusters, cluster A, cluster B, and cluster C. More or less, this represents 3 major haplotype types. There are loads and loads of haplotypes here, but those haplotypes fall into 3 groups. Cluster 1, cluster 2, and cluster 3. And then this is the trait values for each of those clusters. This doesn't come out very, very large, so you can't see it very well, but you've got cluster A, cluster B, and cluster C. Here you've got grain, this is the arsenic level in alternate wetting and drying grain year 1. So you can see cluster A is low, cluster C is high. In alternate wetting and drying year 2, cluster C is high. In continuously flooding, cluster C is high. So the story is the same. So we think that cluster A is low, cluster C, which is really common, is high. But what we can do is we can look at where those haplotypes come from. especially useful is the 3000 Genomes project, where you can download a sequence or SNPs from 3000 genomes, and we can use that to identify that cluster A haplotype comes from japonicas, whereas the C haplotype comes from indicas. It's a really, really important concept to appreciate. If you look for the A haplotype in indicas, almost none of them. There's only a 3000 genomes project, which has got about 1,500 indicas. I don't think there's more than about 10 that have got the japonica allele of land races. You do the same with the japonicas, and you do not find the C cluster. This is a type of allele, and they're quite common in rice, which are specific to the japonica indica subspecies split. And what that means is if you use a japonica population or an indica population to try and detect this QTL, you'll never find it. But interesting is, the thing is that this polymorphism, these alleles are reasonably distributed in the AUS panel. So there are enough of the A cluster in AUS for you to detect this QTL. So the other thing we did is, in addition to the AUS panel, we sequenced some modern Bangladeshi cultivars. So these are 9 modern Bangladeshi cultivars we sequenced at the same time. And we can see how these alleles are distributed within those Bangladeshi cultivars. So what I've done here is I've shown you frequency distribution of grain arsenic measured in the population under continuous flooding in year 1. And each of these numbers represents one of these modern varieties of Bangladeshi cultivars, and the arrows tell you which allele they have. And luckily, most of them, by fluke, I would imagine, have the japonica allele, because they are indica genotypes, but they have the japonica allele for this arsenic QTL, whereas some of them have the indica allele, only 2 of them. I'll give you one other example, and I'll go through this very quickly because I'm aware of the time. So we have another QTL here. I won't do all of the steps that I showed you before, but just the same principle, showing you how different alleles are distributed within modern Bangladeshi cultivars. For this QTL on chromosome 5, we recognize 4 different haplotypes, or major sort of clusters of haplotypes. Cluster B has low grain arsenic. Cluster D is particularly high. But the interesting thing about this QTL is that none of the modern varieties have the low grain arsenic cluster. And we think that's really important because that gives a really strong opportunity for breeding with this haplotype to reduce the arsenic level in grain by about 10%. Nothing more than that, but it would reduce it by 10%. I want to say just one more thing about this chromosome 3. I'll just come back to it. This QTL on chromosome 3, if we actually look what the gene is responsible for that, we know what the gene is. We can use a genome web browser to look at all the genes underneath that QTL, and I'm just highlighting all the genes under this part. So the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 genes under this small region. But really interesting is one of these genes, although it's annotated as an expressed protein, it really is a gene called LSI2, low silicon 2. It's known to be a transporter of silicon and arsenite, and a mutant of that gene reduces grain arsenic by 30%. But that mutant could never be used in breeding because the plants that are mutated for this gene have very poor silicon uptake, and they are very poor plants. If you click on that on the website, you'll be taken to the annotation page for that project, and it says expressed protein here. But if you look further on the annotation of that gene, you see down here it says arsenite transport protein. So it's actually a limitation of the quality of the annotation of rice, that it doesn't give you all the information in the simple annotation. Sometimes the annotation is wrong. In the interest of time, I think I will skip this list section on alignment. It's an interesting addition to what we're trying to do, highlighting the fact that Aus, being a subgroup of rice, is not very similar to Nippon Bari, which is the gold standard reference. You remember when I said when we developed our markers for the BAP population, we aligned it to Nippon Bari reference, and Nippon Bari is a temperature japonica. And that means that the temperature japonica is not the ideal cultivar to use as a reference. So we actually had a look at what would happen if we tried to align it to another reference, which is called DJ123. And I think it's an Indian Aus cultivar. That has been sequenced. The sequence is in thousands and thousands of bits, so it's not a very good reference. I could show you some of that data, but I'm going to skip over it. So these are slides I'm skipping over. Quite an interesting story, but I'm going to skip over that. And just because I want to bring this to a close, I'll say what else we're doing. I have a student, me and Gareth have a student, Kai-Jin, who's working on salt tolerance. So using this population to look at salt tolerance, both in hydroponics and in soil. She's got some really excellent results. QTLs on top of known candidate genes, QTLs on top of some really interesting candidate genes which deserve further study. Ayatunde is another PhD student we have working on cadmium tolerance. So he measures them in hydroponics, measures cadmium uptake and the effect of cadmium on growth. And he gets really, really nice QTLs on top of, sometimes on top of known candidate genes, sometimes on top of new but very good candidate genes. The bat population is being used was being used in the NEWS project, nitrogen efficiency of whole cropping systems. And this is Naraj from the Indian Institute of Rice Research and Anandan from the National Rice Research Institute. They both grow in the population. This is, for example, Anandan is very interested in using it to measure root traits. And he has, for example, detected some very interesting QTLs for, this is deep roots. So he's using the population for that. But now the population is being used in the successor of the NEWS project, which is called the South Asia Nitrogen Hub, where the bat population is being grown at NRI and IIRR. In addition to that, it's being grown now in Biri, in the Bangladesh Rice Research Institute and the University of Agriculture, Faisalabad, under 2 different nitrogen regimes, and we're hoping to use it to look at nitrogen use, efficiency, QTLs. And I think this will be more or less my last slide to sort of summarize where I think we have achieved. I think the BAP reveals QTLs which are not detectable in other populations because the distribution of alleles in Aus is different to the distribution of alleles in other populations. So alleles that are very rare in Indica or Japonica, for example, are not necessarily rare in Aus. So we have interesting trait diversity, but we also have this interesting allele diversity and distribution. We have detected a very large number of QTLs. We have a bottleneck. So we have a challenge here. The bioinformatics to go from those QTLs to publishing an interesting story about good quality candidate genes and takes a long, long time. So we've done the arsenic, we've submitted the cadmium one, we've got someone working on the iron, but we've got others, zinc, sodium. Don't really know how we're going to get it all published. So we need an automated pipeline to list QTLs, list genes underneath QTLs. At the moment, we do this all by hand, do an improved annotation, because at the moment that's not really good enough. revealed genetic variation. Interestingly, we tried to write a grant proposal a while ago with Assam Agricultural University to try and advance this, but we were not successful in getting that funding. We've identified a great number of candidate genes for many traits, and we need a pipeline to test them. And what we'd really like to do is to use CRISPR as that pipeline. We now have loads and loads and loads of excellent candidate genes. CRISPR offers an opportunity to test them relatively quickly. We haven't got anywhere with that yet. We've tried writing grant proposals to get money to do CRISPR, and so far we haven't succeeded. It seems more difficult to get money in the UK than it ever used to be. So we're still hoping. And that's the last slide. I often finish on this because I suppose my justification, my motivation for doing this is the hope that, so especially we can do something that is relevant to some poor farmers. And I guess in a trip to around Bangladesh, a lot of the farmers are really rather poor. These are the kind of people that I'd like to help. And I think our work on arsenic is getting there now. So that's it. I would like to stop and stop sharing my screen and see what happens now.
Vidyut, you are not audible. Vidyut, you are not audible.
Am I audible now?
Yes, yes, sir. Yes, sir. Yeah.
Okay. Yep. So thank you, Adam, for your excellent presentation. I'm sure participants were highly benefited, because numbers of messages I have seen that they have already expressed their happiness attending your webinar, and you have explained your approaches to identify some important genes, candidate genes. And now some people have already raised questions, how they can be used for their applications, especially for the arsenic, tolerance. I have seen some questions, So you can go to the question answer, portion, and then you can identify the questions and you can answer. Thank you very much.
Before that, Adam,
Yeah.
thank you very much for a very informative speech. I think given more time, you could have only spoken for at least 1 hour more. However, time is, of course, of limitation. And I would like to ask you one thing. As you yourself said that you have compared your GPS data with Nippon Bade, but you have also tried to do it with host reference, that is DJ123. And what kind of difference do you get?
The idea about comparing with DJ123 is that essentially the logic is like this. There will be parts of the genome of Ausis which is not in Nippon Bari. So we identified from the DJ123 reference parts of sequence, parts of contigs which were not in Nippon Bari, and then we did GWAS just on those. And we find some significant GWAS. The challenge we have when we get peaks on this parts of the DJ123 genome is the annotation of that, because we don't know what's underneath there. At the time we did that, which was about 3 years ago, there was no annotation. We ought to come back to it. We ought to do it again, because the annotation of both DJ123 and now N22 has been fully annotated now. It wasn't 3 years ago. So doing it again with a fully annotated AUS reference would be a very interesting exercise. But for example, we think we found significant association underneath PUP1. PUP1 is the phosphorus tolerance QTL, which is in Kazalat, an AUS cultivar, which is not in Nippon Bari. So Yeah. So look, there's quite some questions here, and I guess they're going in reverse order. So the one at the bottom, no, the one that I can't see what order they're in. There's a question about GAPIT and high p-values. I don't know how to answer that. I don't know the answer to that one.
Okay.
Is Alex there?
I'm going to skip that one. So I'll take this one here by Mamta. Great work. Does arsenic play an important role in rice growth and development? So the issue about rice is arsenic is it's toxic. It's toxic to humans because it causes cancer. It's toxic to plants because it interferes with, probably because it interferes with phosphorus metabolism. And arsenic in soils is definitely phytotoxic. We think that's somewhat less of a problem because the level of arsenic to be a problem for rice growth is quite a lot higher than it would need to be a problem for human consumption. Having said that, and I think more research probably needs to be done on it, having said that, I think there are areas where there is quite a lot of arsenic in rice fields where you may be getting to levels of significant phytotoxicity. But you would reduce that phytotoxicity, by the way, quite substantially if you grew the plants aerobically, because the arsenic is much less available to the plant. Right, I've got this question from Pradipta. Whether this QTL can be utilized for the development of arsenic-tolerant cultivars. So, as I say, the main focus of the work I presented is for low-grain arsenic, and what I suppose I wanted to highlight is some of the QTL. Definitely look as if in reducing grain arsenic. LSI2 and the 105 that I showed you, and there are others. They could be used for breeding. The person doing the breeding must check whether or not which allele elite variety has. I showed you before, most Bangladesh have the low-grain arsenic allele for LSI2. So there would be no point doing breeding with that one for most cultivars in Bangladesh. But for the other one, there would. So I'm just hoping now we've published this, that people involved in breeding for low-grain arsenic will use that to start breeding. There's one caveat. This is only one field site. This 2 years and 2 different treatments. But it would be nice to know whether or not this works in another field site. Okay, and I've got another one here from Sanhani. A very interesting topic. Can you please state the ways of validating GWAS results? Yes, that's actually a marvelous question because whenever you get GWAS QTL, it's exactly the same as when 20 years ago I used to get a QTL using conventional mapping. You want to know whether or not it's real. And there are multiple ways in which you can either give yourself greater confidence or finally prove. So the only way really to prove whether or not your guess about what is a good candidate gene is to do some forward genetics. So reverse genetics. So knock the gene out, or ideally use CRISPR to change alleles. The perfect result would be to use CRISPR to change alleles. I don't think that's even perfected yet in rice, using CRISPR to swap alleles. But some kind of transgenic or CRISPR approach would be the perfect way to prove. But you don't necessarily have to prove, in my opinion, before you could use it in breeding, as long as you've got really good confidence. And that confidence is gained by having a QTL which is repeatable. In other words, you don't just find it once, but you find it more than once, or somebody else has found it, or you've got a good gene. You've got a gene underneath that QTL which somebody has already shown is involved in cadmium. But if you have a situation where you find a QTL, no one else has found it because they've not looked in the right population, for example, or no one's measured that trait, and there are no good candidate genes, then the only way to validate would be to start knocking genes out or to start swapping alleles, which is a lot of work, but CRISPR should make it easy.
I think in your last slide, that was shown for your candidate genes. Now you know you have a plan to go for CRISPR to validate those genes.
We don't have a plan to go for CRISPR because we don't have the funds to do it. So we will do that once we have the plain truth is, in Aberdeen, we don't have expertise in transgenics. So we don't have anyone here who can do CRISPR. So we would need to do that through a project which we fund somebody running a CRISPR facility. So we've tried to get money. I have discussed with yourselves in the past trying to collect. So that would be the only other way to do it through collaboration. Right, I've got another question. Where am I? Here. This is Mahamaya. What type of population are best for GWAS in rice? Right, that simply depends on what you're trying to achieve. So it's a complicated question. We had a very logical reason for focusing on ours. It was because the region that we were interested in, where the arsenic problem is strong, is northeast India and Bangladesh, which is the same region where this subgroup of ours comes from. So the other reason is by working within one subgroup, you eliminate some of the problems associated with population structure. But you need to be a little bit careful because for sure you need to choose a population in which there is sufficient variation to do GWAS. I'm not sure that's likely to be a problem because there are so much genetic variation within rice. It's not difficult to find markers, 2 million markers. So people are working on 19 million markers. So I don't think getting markers is a problem. Trait variation, you need to be confident you're going to get trait variation. So there are loads of really nice studies on populations of just japonicas or just indicas, or you can go for a population which covers everything. But to do that, like the rice diversity panel, to do that, the expectation really is to do good quality GWAS, you need about 200 genotypes. So if you have a population, a global population, which has all the subgroups of rice, you really need 200 accessions from every single subgroup. There are 5 subgroups, so that means a population of about 1,000. So the rice diversity panel has an expanded set called the rice diversity panel 2, which has 400 accessions. If you can work on a population like that, that's fantastic. But we never had the resources to work on a population that size. And then I got a question from Debajit. Yes, so what about developing a haplotype based near-isogenic line for developing cultivars? Yes, well, I think that's essentially is the approach you would use in breeding. With the information that we've got, or further exploring the 3000 Genomes project, you should be able to identify accessions, available accessions, which have the low arsenic haplotypes. And then you can develop from that haplotype markers, which you can ensure that you use in your breeding to make sure that that haplotype gets across in your breeding program. I don't know if that really answers the question.
Yeah.
And we have
any more questions from participants?
One from Sushil. What is the lowest concentration of arsenic linked to the mapped QTLs? What's the threshold concentration? not sure I understand the question, but what I would say is, first of all, for grain arsenic and safety, there is no safe threshold for a class 1 carcinogen, but the Indian government, the Chinese government has put a limit of 150 micrograms per gram inorganic arsenic in rice. And many of our samples in that, most of the samples in our study were over that. And the QTLs that I'm describing in our paper, because you see there were a load of QTLs, they tend to reduce the arsenic by about 10%. None are bigger than 10%. So if you want to really reduce it substantially by 50%, you'd have to use multiple QTLs. I think that's it.
Yeah.
So Adam, I have one
any
question.
more question?
I have
Okay,
one
Manoj,
question.
please.
It's Manoj.
Please.
Adam, it's
Please.
Manoj from NIPJ New Delhi.
Oh yeah.
Okay.
See you.
Yeah. So Adam, you have done a lot of work on arsenic stress by using the Indian cultivars. So which QTLs are consistent in all of your analysis? How many QTLs are consistent?
From the study I presented, we have 6 QTLs which are consistent across our experiment. So that's as far as I could probably say. So we have been working on arsenic for a long time. There are a number of features about arsenic which make it complicated in breeding. Which is why we did struggle. One is that there is big QTL bioenvironment interaction, as evidenced from that slide I showed you from the rice diversity panel. Another one is that there is quite considerable epistasis. We got some evidence that there's quite considerable epistasis. So in terms of Indian varieties, I don't know, but if you look at that paper, you should be able to pick up those cultivars, or at least from the data that we present in the paper, you should be able to pick up cultivars which have the low arsenic allele for any QTL. Certainly for the ones that we presented there, it wouldn't be difficult. I don't know if that...
Any gene you think that would be important out of the 6 QTL?
Say that again?
Any gene or candidate gene you have identified within the 6 QTL region?
That essentially the japonica allele of LSI2 is a no-brainer. That should be in any cultivar that you're growing in high arsenic region. It's an absolute no-brainer. As I said in my talk, just by luck, I guess, most Bangladeshi cultivars seem to have the japonica allele. So that's the only one we can identify for sure with a high degree of confidence to a gene level. There's an ABC transporter, I think we also highlight in that paper, but it would be nice to do CRISPR on that to really be sure. I can't remember, Manoj.
Yeah, but do you think that knocking out one gene will solve the problem of arsenic stress, because it is QTL and then environmental interaction is also there?
No, you can't knock the gene out. So if you knock out LSI2, the one that we identified on chromosome 3, if you knock that gene out, you get a 30% reduction in arsenic.
Okay, then it's a great major QTL.
No, it's not good because you also reduce silicon and you get a plant which is much less healthy.
Okay.
So you can't knock the gene out. I think it would be naive to assume that you could just knock the gene out. What we're trying to achieve here is identify a superior allele from natural allelic variation. So if you use the japonica allele for LSI2, you'll get 10% lower, not 30%, but 10%. But you won't introduce an allele which will have a major impact on other aspects of the way the plant grows.
Okay.
There is no silver bullet here for grain arsenic.
Thanks.
Okay.
Thanks, Adam. Nice
yeah,
presentation.
there is one more question from MD Patil. Adam, could you see that question? from MD
Yes,
Patil.
okay.
Yep. If there is any
Yeah, so linkage drag. That is the beauty of using marker-assisted breeding. If you make a cross with a donor of your resistant, your low arsenic gene, you make a cross. You have markers close to it or on it. You have markers some distance, small distance away. You can use those markers to completely eliminate linkage drag, but you will have to assess a large number of progenies. That is the beauty of marker-assisted selection as a tool to eliminate linkage drag, which is an inevitable consequence of crossing. Does that help?
Okay. So, is there any more question? If not, Dr. Modi,
Yeah,
should we wind up?
the point that I was trying to
Somebody,
make,
Raman is having a question. Okay, Raman, please.
Adam, so how do you reduce the number of SNP? Because you have number of so many SNPs, and for your grouping pattern or for your structure or some other Bayesian analysis, how you reduce the number of QTL to 500 or 300? What is the basis in spite of having 2 lakhs or 1 lakhs of QTLs?
SNPs.
Yes, SNPs, yes.
You're vaguely familiar with the approach we actually use, but what we do is we have a Tony, who does our bioinformatics, has written a small program called Extract. So that allows us to specify a region within the population, and then we can extract all of the SNPs within that region. So if you just choose a gene, you can extract all the SNPs in a gene. That tends to give you something like 20 or 30 or 50 SNPs. Or if you want a 200 kb region, then you're looking more like 500 SNPs. But you can easily, with a command line, extract SNPs in any region. I'm not familiar with Tassel. When we started this program, doing this, Tassel was not able to handle 2 million SNPs, so we didn't use Tassel. And Tassel slowly improved in terms of the size of populations it can handle. And I don't know whether or not Tassel offers an opportunity to extract SNPs in that way. It may do. I'm sure there are other ways. But basically, you're just extracting data from a massive file which contains all the SNP data.
Yeah, okay, Adam, thank you.
Okay. Okay, Raman, thank you.
Thank you.
And Dr. Modi,
the point I was trying to
anything
make,
more?
Adam has already mentioned that because silicon uptake and arsenic uptake are overlying each other, they're both mechanisms. So if you try to reduce arsenic uptake too much, your plant may not have the rigidity that you need. There'll be a lot of losing, lot of weak plants and all that. Adam already mentioned that. So thank you very much, Adam. Very nice talk, very informative.
Yeah, it's very nice
Okay.
to be
Thank
able
you.
to interact with you again.
Yeah. Thank you all participants. Thanks to Dr. Adam for being with us and for your excellent, speech today. And our next webinar will be on 26th morning, that is at 11 a.m., which will be delivered by one of our collaborators in Organic Agriculture Group, with Organic Agricultural Group, that is Dr. Deshpande from Pune, who was earlier in NCL, National Chemical Laboratory, Pune, and now after retirement, he has opened a company. So you'll be listening to him that how he developed various bioformulations, especially the biopesticides and biofertilizers, and how he has now gone into commercializations of his own products. So I'm sure you'll be enjoying. So with this, I'm winding up here for today. Thank you all.
Thank you, everyone. Thank you, participants.